Good morning and welcome to the 31st meeting of the IPAC meeting. I'm Mike Kaczynski. I'll be managing today's meeting. Periodically, you'll hear me jump in case we have any technical issues. We do have a great agenda today. This is a public meeting. So I'm going to get this kicked off right away and hand it over to our chair, Dr. David Peden. Dr. Peden, take it away.
Well, thank you, and I thank all the members of the committee and all the FDA staff who are taking time to undergo this, to participate in the meeting. The major agenda item is the discussion and review of the laboratory of immunobiology, and also the and then we will ask members to review and to discuss and either approve or amend the site visit report that for the site visit we conducted some months ago. So with that, I call this meeting to order. The next item, I don't have very many other opening remarks besides that, so I think, Kathleen, we can catch up on time by moving on to the administrative announcements, roll call, etc., and proceed from there.
That sounds great. Thank you, Dr. Peden. My name is Kathleen Hayes, and it's my pleasure to serve as a designated federal officer for today's 31st APAC meeting. And on behalf of the FDA Center for Biologics Evaluation and Research and the committee, I just would like to welcome everybody to today's virtual meeting. As Dr. Peden mentioned, today will be to hear overview presentations of the research programs in the Laboratory of Immunobiochemistry within the Division of Bacterial, Parasitic, and Allergenic Products. And today's meeting topic was described in the Federal Register notice that was published on September 9th, 2021. For any media or press-related questions, you may contact FDA's Office of Media Affairs at fdaoma@fda.hhs.gov. And the transcriptionist for today's meeting is Ms. Linda Giles. So we're going to begin our meeting by taking a roll call of the committee members. And when it's your turn, please just turn on your video camera and unmute your phone, and then state your first and last name, your expertise, and your organization. And when finished, please turn off your camera and we'll proceed to the next person. So we will begin with Dr. Peden. If we can pull up our roll call slide here. There we go. Go ahead, Dr. Peden.
Good morning. I'm Dave Peden. I'm with the University of North Carolina at Chapel Hill, and my expertise is in asthma, pediatric allergy, and impact of environmental agents on allergic and airway disease.
Thank you, Dr. Peden. Dr. Assad?
Good morning. I'm Amal Assad from Cincinnati Children's Hospital Medical Center and the University of Cincinnati. I am an allergist immunologist, so my experience extends over the spectrum of allergy immunology, but specifically I have larger experience in food allergy. Thanks for having me.
Thank you, Dr. Assad. Dr. Davis? You might be muted, Dr. Davis. Can you check your phone?
Oh, thank you. I thought I pressed that. Hello, my name is Dr. Carla Davis, and I am at Baylor College of Medicine, Texas Children's Hospital. I'm teacher of the Division of Immunology, Allergy, and Retrovirology. So my interests are in food allergy as well as the immunology of allergic responses.
Thank you. Dr. Dykowitz?
Hey, good morning. I'm Dr. Mark Dykowitz, Chief of Allergy and Immunology at St. Louis University. Of note, been very involved with practice guidelines, including lead editor of the 2020 Joint Task Force National Rhinitis Guidelines, looking at diagnosis and treatment of rhinitis.
Thank you. Dr. Greenberger? You might be muted, Dr. Greenberger. Go ahead. I think you're unmuted now. I can hear you.
Paul Greenberger, Northwestern University Feinberg School of Medicine. I'm in the Department of Medicine. I run the CLIA-certified lab in our medical school for testing for allergic bronchopulmonary aspergillosis, hypersensitive pneumonitis, and occupational immunologic lung diseases, and have other interests in adult allergy, immunology, asthma, anaphylaxis, and some adult immunodeficiency.
Thank you. Dr. Keat?
Hi, I'm Corinne Keat. I'm at the University of North Carolina. I'm a pediatric allergist with an interest in food allergy and asthma.
Thank you, Dr. Malecki.
Hi, good morning. My name is Sohaila Maliki. I'm with the U.S. Department of Agriculture, and I study the structure, function, and biochemistry of food allergens. I'm also working on developing new diagnostics and therapeutics for nut and tree nut allergies.
Thank you. I think we just received a note from Dr. Nolte. He's in attendance, but I think he's having some trouble connecting his audio, so I'll come back to him. Dr. Portnoy?
Good morning. I'm Dr. Jay Portnoy. I'm a professor of pediatrics at the University of Missouri Kansas City School of Medicine. I'm an allergist immunologist at Children's Mercy Hospital in Kansas City. My area of specialty is environmental assessment and interventions, more recently food allergy and diagnostic testing. Thank you.
Thank you, Dr. Portnoy. Dr. Woodfolk?
Good morning. I'm Judith Woodfolk, a professor of medicine and microbiology at the Department of Medicine, University of Virginia School of Medicine, and I bring expertise in allergy and immunology with a focus on adaptive immunity to food and inhalant allergens and rhinovirus.
Thank you, Dr. Woodfolk. I believe my camera is frozen, so I'm just going to go ahead and so thank you for your introductions. I also just wanted to acknowledge the contributions of a few other members of the DSAC team, including our director and co-DFO for today's meeting, Dr. Prabhakar Atreya, Ms. Monique Hill, and Ms. Karen Thomas, along with Ms. Christina Vert, also assisted in preparing for this meeting, and I would like to express my thanks to Mr. Mike Kaczynski for facilitating. I also wanted to acknowledge CBER leadership and management, including Dr. Marks, Dr. Witten, Dr. Elkins, Dr. Gruber, Dr. Kraus, Dr. Chumakov, Dr. Slater, and Dr. Burns, some of whom will be joining the meeting later today, and others who will be providing overview presentations shortly. So before we begin reading the conflict of interest statement, I just wanted to mention a few housekeeping items. For member speakers and FDA staff, please just remember to keep yourself on mute unless you're speaking to minimize feedback. And if you've raised your hand and are called upon to speak by the chair, Dr. Peden, just as a reminder to speak slowly and clearly so that your comments are accurately recorded for transcription and captioning. So I will now proceed with reading the conflict of interest statement. The Food and Drug Administration is convening virtually today, October 28, 2021, for the 31st meeting of the Allergenic Products Advisory Committee, APAC, under the authority of the Federal Advisory Committee Act of 1972. Today, the APAC committee will meet in an open session to hear overview presentations of the intramural research program in the Laboratory of Immunobiochemistry in the Division of Bacterial, Parasitic, and Allergenic Products, offices of the Vaccine Research and Review Center for Biologics Evaluation and Research. Per agency guidance, this topic is determined to be non-particular matters which would have no impact on outside financial interests, hence no affected firms are identified and members are not screened for this topic. Today's meeting will be closed from 12:20 to 1:20 p.m. to permit discussions where disclosure would constitute a clearly unwarranted invasion of personal privacy. Dr. Peden is serving as the chair for both the open and closed session for this meeting. And the following information on the status of this committee's compliance with federal ethics and conflict of interest laws, including but not limited to 18 U.S.C. Section 208, is being provided to participants in today's meeting and to the public. With the exception of the industry representative member, all standing and temporary voting or temporary non-voting members of APAC are appointed special government employees or are regular government employees from other agencies and are subject to federal conflict of interest laws and regulations. Given that the topic of this meeting is determined to be non-particular matters, it has also been determined that the overview and updates of this meeting present no actual or appearance of financial conflicts of interest. Dr. Hendrik Nolte of Merck serves as the industry representative to this committee. Industry representatives act on behalf of all related industry and bring general industry perspective to the committee. However, industry representatives are not appointed as special government employees and serve as non-voting members of the committee. They are not authorized to attend any closed sessions if held. Therefore, industry representatives are expected to leave when the open session ends. Dr. J. Portnoy is serving as a consumer representative for this committee. Consumer representatives are appointed special government employees and are screened and cleared prior to their participation in the meeting. They are voting members of the committee and hence do have voting privileges, and they do participate in closed sessions if they are held. FDA encourages all meeting participants, members, or consultants, including open public hearing speakers, to advise the DFO and committee if they have realized any financial, professional, or regulatory relationships with any of the topics or individuals being discussed today that were not previously disclosed, and recuse themselves from committee discussion, and their absence will be noted for the record. This concludes my reading of the conflict of interest statement for the public record, and at this time I would like to hand it back over to Dr. Peden. Thank you.
Thank you for proceeding with the announcements and conflict of interest statement. The next item on our agenda is the overview of the CBER research programs. I'd like to introduce Dr. Karen Elkins, the Acting Associate Director for Research for the FDA, who will be making that presentation. Dr. Elkins, if you would turn on your camera and unmute your phone, and please proceed.
Good morning. Can you hear me now? Maybe not. Ah, yes, you can hear me now. I have a new headset, so I'm actually getting a lot of feedback myself. Let me see if I can improve that before I distract myself.
We can hear you. We can hear you okay.
Okay, good. As long as you're not hearing the feedback that I am. That's all that really matters. The volume and do a little bit better. All right, so I'd just like to give you a brief overview of the CBER research program in general as some context for your deliberations and review of this report today. And we very much appreciate your input in the review of the research program, another very important function of our advisory committee, in addition to looking at our licensed products. So, Mike, I am looking for the usual buttons. Aha. There we go. Maybe they're coming along now to be able to move the slide. And I'm still not seeing
They should be at the bottom of the screen, the little arrows at the bottom.
There they finally. Okay, got it. The screen did something funny. Apologies. So as I'm sure you all know by virtue of your service on this committee, CBER regulates a wide range of complex biologics, including blood products, a suite of cell and gene therapies, vaccines, live biotherapeutic products, and of course the products close to this committee's heart, allergenic. And this in itself asks for a lot of scientific expertise. The word cloud is drawn from our entries into our annual report process and gives you some idea of the propensity of our scientific reviewers and regulators to lean toward biological, immunological, bacteriological, and important at the moment, virological expertise. Our research program is an explicit part of our strategic goal. Goal number 2 calls out conducting research to address challenges in the development and regulatory evaluation of medical products. And we have had this way of doing business for pushing 75 years now. and that is we are comprised of researcher reviewers. We have laboratories that look a bit like academic laboratories in the sense that they're led by PIs who propose investigator-initiated research in the context of their regulatory review assignment. And so our research programs, because they are investigator-initiated in part, cover a range of what might be described from basic research studies to very targeted studies that are very directly related to our regulated product portfolio. We think this way of doing business ensures a state-of-the-art understanding of techniques that are the source of data in regulatory decisions, that it keeps us a cadre of scientists who are efficient, effective, and credible in their review work, and that the approach fosters decisions based on sound science. So our research and our review are integrally linked. The research staff are primarily assigned as part of regulatory teams to focus on product-related issues. In the jargon, the chemistry, manufacturing, and control reviewers. And they are tasked with looking in our regulatory submissions at the scientific rationale and data that underpins the proof of concept for taking the leap of putting a new product into human beings. They focus on the product, the production techniques themselves, and the nature of the resulting product, how that product is quality control tested, and if there are assays from clinical specimens resulting from clinical trials, laboratory-based assays, they also review and evaluate those. It's important for you to note that our research staff spends a substantial amount of time, therefore, functioning as regulatory product reviewers in the context of an overall team. The percentage of time varies, but it can be up to about 50% for PI, somewhat less for more junior or newer staff that are still on the learning curve, but it's substantial. This review is done in the context of a team that is also comprised of a regulatory project manager that provides oversight of the entire process, a farm tox reviewer, a clinical reviewer who naturally focuses on the clinical trial design and the resulting clinical data, and a statistical reviewer who helps us put it all together. So our researchers are going back and forth between being a member of a large regulatory review team and directing the research in their laboratories that is the subject of today's review. We think this approach ensures that science is applied throughout the life cycle of a product, from its start as an idea through the challenges it may present in regulating and developing it, to understanding risk benefits, to ending up as a licensed product that impacts public health. Our labs take place on the White Oak campus in Silver Spring, Maryland, where you're not meeting. We hope to see everybody in a real meeting one of these days again. We have two large buildings that comprise about 450,000 square feet and encompass about 150 labs. They range from BSL-1 level work all the way up to BSL-3 level work. And at present, our research staff is a little more than 500 people. On campus, we have a number of core facilities that provide on-site technologies for flow cytometry, imaging, bioinformatics, and all kinds of molecular biology. We also have a state-of-the-art vivarium that now works with, I think at last count, 12 to 13 different animal species, going from rodents up to ferrets and primates. Our research activities result in a number of external collaborations. This draws from our most recent data that illustrates that most of our collaborations, as you might expect, are with academia and other components of FDA and the government, such as NIH, our neighbors down the road. Relatively few, but nonetheless a substantial number of collaborations are with regulated industry. As you might imagine, these are carefully monitored and managed, but that can happen, and we hope that those kinds of public-private partnerships also facilitate product development. And our external collaborations rise to the level of formal agreements for a number of mechanisms, contracts, some external grants, technology transfer, intellectual property results, and the usual slate of material. So we think this process has a number of benefits. It prepares us for future products that are innovative and unusual, and for a variety of public health challenges, while facilitating the recruitment and retention of a cadre of highly trained scientists that have the expertise necessary to quickly and effectively review regulatory submissions. And I think we are living the example of this benefit right now. Our virologists, specialists were in place, ready to go when the pandemic hit. The research activities can, in some cases, directly develop data and tools for the development of classes and products, besides providing foundational knowledge. And we always seek to fill knowledge gaps that will aid product development, particularly in informing policy and regulatory decision making. Our research programs are evaluated in a number of ways. Annually, all investigators enter progress and next year plans into an annual report mechanism that is then reviewed by lab chiefs, division, and office management all the way up the chain, with feedback going back and forth about the progress and plans for the programs. We have a formal horizon scanning program that takes place every 4 years that involves office and center staff. The new projects get particular scrutiny through all levels of supervisory at the time they're initiated, and new projects may come along, for example, in response to COVID-19, or simply new ideas that arise out of investigators' personal horizon scanning of what's coming down the pike and needs to be addressed. And there's an internal intramural review process to initiate new projects. And then we have external review. And that is the subject of today's discussion, where we ask subject matter experts to form a committee in association with the relevant standing advisory committee to come together for a review of a particular selection of lab programs, usually several PIs in one shot. We have tried to stage these every 4 years. That schedule has been slipping a little bit in the last year or two, but the intent is to review a period of time that can demonstrate progress without going too long between getting feedback from you all. So the site visit in question for today's was held in late April, and we asked the site visitors to evaluate the work and progress of the lab investigators in question according to the usual criteria, things like dissemination, what has resulted in the way of presentations, publications, or tech transfer development, whether the work itself is mission relevant. We understand that you might not know the details of what mission relevance means, but we are sure that you can judge it in a general way. And we are particularly interested in the opinion of whether the work in general is impactful, that is, whether it is reaching out to the scientific community and being taken up there and being useful to all of our stakeholders. So that results in the site visit report that you had to review. The draft report was prepared shortly after the site visit itself in late April and then has been distributed for your further review. There are 3 possible outcomes of today's activities. First, you can accept the report and amend the report, or you can reject the report and send it back to the site visit team for further work. Once the report is final, it is used internally in a number of ways. Its contents, the contents applicable to each program, goes to the PIs. I assure you that they take your criticism to heart and use it to improve their research programs. Supervisors use it to discuss the nature of the research programs for internal review, and supervisors and management use the outcome to discuss allocation of resources. Money, space, and people can all be impacted by site visit reports in conjunction with all of our other research review approaches as well. So with that, I'd like to thank you very, very much for your input into this. You are a big part of what ensures our critical mass to maintain our high-quality research programs and to stay on target with our regulatory mission. very much appreciate your comments, your insight. We take all of it, and I'm happy to answer questions too.
All right. Thank you, Dr. Elkins. And at this time, I invite members of the committee, if they have questions for Dr. Elkins, we have about 5 minutes allocated for that. If you're interested in asking a question, please raise your hand digitally.
And just a reminder to everyone to raise your hand, dead center, right above Karen is their that little caricature with their hand up. So go ahead and click on that. Otherwise, like I said, and then to unmute. But in the meantime, Dr. Peden, do you have any questions for Dr. Elkins to kick it off? And if you could, turn your camera on too.
Yeah, well, I was keeping my camera off to allow others to move in, but well, first of all, thank you for that very good overview, and you may have actually satisfied most questions in terms of what the role of the review is, et cetera. One general observation I think the site visit team did have, and I'll pose it to you. Many of the observations that the site visit team made for the investigators involved access to clinical samples, involved access to the clinical populations, and realizing that COVID kind of disrupted a lot of our normal work. I just wonder if there's what support or what infrastructure is there to help facilitate that for investigators.
Could you say the last part again? I'm not sure specifically.
I just wonder what organizational support there may be for to help facilitate access to clinical samples or clinical populations in the context, given that you don't have the same kind of campus as, say, your neighbors to, I think, the west of you.
Right. Well, what we do have is a wonderful tech transfer officer, Misha Narayanan, who organizes and creates all necessary agreements between other parties, and that's usually the starting point of receiving clinical samples. Nisha, in turn, is a subset of Big FDA's tech transfer office, which also has the capability to facilitate receipt and appropriate protection and agreements regarding all clinical samples. I do rely on the investigators to start that process to identify sources and to bring to our attention options that we may explore, but we have both the expertise and the personnel to facilitate just about any kind of agreement we can.
Okay, thank you. Before we move on, are there any other questions from any other members of the committee? Well, if not, thank you, Dr. Elkins, for that for the overview. And
you have one from Dr. Portnoy. Dr. Portnoy has a hand up.
Yeah, I just had one question. Thank you for that presentation. Very interesting. My question, we're going to be reviewing the site visits for the different laboratories in the FDA for the allergenic products. And I guess my question is, how much effort are these investigators expected to put into regulatory activities as opposed to original research activities? What is the total scope of their work, and how do we take that into account when evaluating the productivity of the labs?
Yeah, that's an important point. So the principal investigators are usually expected to put on the order of 50% of their time into regulatory review activities. That is 50% plus or minus 25. The regulatory workload can vary according to first what sponsors submit, so they have the control on that side of it. And also where a PI is in the learning curve. So new PIs start off by learning and devoting 10 to 20% or so of their time to both the learning and the conducting of reviews while they are getting their programs established. And then over time, that expectation gradually increases. The counterbalance to that is that staff scientists permanent but subordinate to a PI also become involved in regulatory review. They carry on the lab activities, but they may spend up to, say, 20 to 25% of their time in regulatory review. And senior technicians who develop an interest and an ability may also contribute to regulatory review. So the entire group is being managed to both keep the research program going as well as contribute to their expertise on the regulatory side. I think it's somewhat analogous to an academic appointment where you have teaching expectations, student expectation, committee work, and the like within the department. The minor catch to that analogy is that hopefully the teaching and the student demands can be somewhat anticipated and scheduled. Regulatory work's a little bit less under our control. When it arrives, but I think we've become fairly adept over the years at helping manage workload across investigators. Big exception being COVID-19 for the vaccine and the convalescent plasma people, but does that help?
Yes, great. Thank you very much.
Okay, if there are, Dr. Malecki, am I seeing that right, that you have a question? I think you're on mute.
Here, I'll help you. You're unmuted now. There you go.
Yeah, I think I accidentally clicked it a couple of times. So just curious, as far as the regulatory, this of their work, as you say, as it arrives, regarding the regulatory work, how, what types of functions do the scientists, do they actually perform laboratory experiments on products that are being regulated? What exactly does that involve? Just not, you don't have to give me detail, but just a kind of an overview.
No,
thank you.
The vast majority of the activity is review of documents submitted by sponsors. So descriptions of product, descriptions of the assay, descriptions of production techniques, and then critiquing that, looking for expectations of what should and shouldn't be happening with generation of a product, how it is quality controlled, and then translating that back into a memo that turns into questions to the sponsor that then a sponsor addresses and gets guidance for how to continue with their product development. We do very little in-house product testing, and that is handled by a separate quality control group that is independent of the research program. There is some exception to that with the current group, and I think Ron is going to tell you a little bit about that in the course of his presentation.
All right, thank you. I don't see any other hands up, and I do want to try to keep us on some semblance of time. We have a fairly full agenda. All right, thank you.
Thank you all for your work today.
The next speaker will be Dr., and forgive my pronunciation, Dr. Konstantin Chumakov, Associate Director for Research for the Office of Vaccines Research and Review, FDA. And Dr. Chumakov, please activate your camera and unmute yourself.
Yes, good morning. Today I will give you an overview of the Office of Vaccines Research and Review, as well as the Division of Bacterial, Parasitic, and Allergenic Products. The products that this office regulates include, of course, as is clear from its title, vaccines, allergenic products, but also live biotherapeutic products, meaning probiotics, as well as fecal microbiome transplantation, as well as phage therapy. The mission of our office is to protect and enhance public health by assuring the availability of safe and effective vaccines, allergenic products, extracts, both diagnostic and therapeutic, and other related products. We do this by involving in three types of activities. First, of course, we review, evaluate, and take appropriate actions on investigational new drug applications, biologics license applications, various amendments, supplements, as well as participating in inspections of manufacturing facilities. Second, we also develop policies and procedures that govern the market review of the product. And finally, we also conduct research related to the development, manufacture, and evaluation of vaccines and other products that we regulate. And the mission, our research mission, is to complement our regulatory review mission by focusing on issues that are related and require immediate clarification. So the organizational chart you see on this slide, and as you may know, we're in a transition because both Dr. Gruber and Dr. Krause are leaving the agency, and Dr. Marks will be serving as an acting office director until the selection of a permanent replacement is made. But nevertheless, the office contains three divisions. A division on the right, a division of vaccines and related products application. Staff in this division is involved in a full-time regulatory review of products and involved in medical clinical review, pharm tox review, managing the entire regulatory process. And divisions on the left, division of viral products and the division of bacterial, parasitic, and allergenic products. This is where staff is involved in both regulatory activity as well as conducting research. This is where our so-called researcher reviewers reside. The goals of our research program are threefold. First, of course, we investigate safety of products, and this includes various aspects of manufacturing process. We develop models and methods, reagents needed to manufacture and evaluate these products. Second aspect of our research program is studies of efficacy. So we develop new assays and reagents needed to assess safety, assess efficacy of products. We study the biomarkers of efficacy and all things related to the effectiveness of the product. And finally, we also work on availability, meaning that we develop different approaches related to manufacturing and all aspects that are not covered by the safety and efficacy, because of course products will do no good if we cannot really manufacture it in an effective way. So why do we think research is particularly important for our products? First aspect is that we have a very strong emphasis on safety, because our products are used in general population, in healthy population, often in children. So of course tolerance for safety is really of paramount importance. So some of our products are very old, and new manufacturing technologies are developing very fast, and we need to keep up with this new innovation. So there is a tremendous scrutiny by the public on what we do and on our products, and the best examples, of course, were provided by this pandemic, where there was a lot of attention in general public to vaccines that we regulate. Of course, our products are important for responding to public health threats, not only pandemic threats, as we all witnessed recently, but also developing antibiotic resistance and emerging other adventitious agents and so on and so forth. But also what's important, I think, is that our research is kept in public domain, and it benefits not only just one company. as would research and industrial in companies would do, but rather benefit the entire line of products, the entire industry, and therefore it benefits American consumers. So we want to keep our research program as broad as possible. Of course, we cannot cover every aspect of the wide variety of products that we regulate, but we want to cover as much as possible. And the reason is that it allows us to quickly respond to new emerging threats. So because we have a very diverse expertise in a variety of different fields. Since we cannot really, our resources are limited, we try to be as collaborative as possible. So we try to keep collaborations with academia, sometimes with industry, just to enhance our ability to address the issues that we need to investigate. So we strongly believe in the investigator-initiated model. So especially since our research staff is involved in regulatory process, that makes it even more effective because our investigators, they really are exposed to the most recent trends in industry. Of course, we want to support only the best research, and we want our researchers to be a part of a broader scientific community and keep links to the with their peers in academia and other institutions. And of course flexible, because I mean we never know what's on the horizon, what's coming our way, and as I said, keeping the broad expertise in the office allows us to rapidly change course, and some programs immediately respond to emerging public health threats. And of course, as Dr. Elkins already mentioned, we employ the researcher-regulator model, meaning that our staff is involved in both activities, and it basically enriches both sides. So, of course, being in the lab and keeping to the research community, general research community, helps us being better regulators and being exposed as a result of our regulatory work to the most current issues that are facing the industry. It allows us to address these issues immediately, and that's why we think that this is a very important way to do research at the agency. So now I will give you a brief overview of the division of bacterial, parasitic, and allergenic products. This is where the lab of immunobiochemistry that you're reviewing today is located. So this division employs 17 principal investigators. They are grouped into 4 labs, and I will give you in a moment a brief description of each lab. So the regulatory research portfolio includes this long list of different bacterial, parasitic products, as well as other products that are regulated by the office. So the first and the biggest lab in the division is the division of the lab of bacterial polysaccharides. It regulates, and you can see it highlighted in blue, invasive bacteria that protective responses to which are based on polysaccharides, as well as Salmonella typhi. The next lab is Laboratory of Respiratory and Special Pathogens that mostly regulates and performs research on non-invasive toxin-producing bacteria, such as Bacillus anthracis and Clostridium botulinum, some of which have importance in terms of counterterrorism activities, as well as Staph aureus. Laboratory of Mucosal Pathogens and Cellular Immunology actually regulates and conducts research on a wide variety of bacteria and other products. And the reason is that this lab actually has some investigators. It was basically a result of a merger of several labs, and now it has a really very, very wide portfolio, starting from Clostridium difficile, intracellular bacteria, enteric bacteria, parasitic malaria vaccines, as well as other emerging products such as Staph aureus, probiotics, FASH therapies, and microbiome-related products such as FMT. And finally, the lab that you are reviewing today, immunobiochemistry, and they regulate just one type of products, allergenic products, but of course you understand that it's very deceptive because there are so many of them, more than 1,000 individual products, that it's a really, really significant workload in terms of regulatory activities as well as research. And as you will see from Dr. Rabin's presentation, this group is also focusing on immunology, on studying innate immunity, interferon responses, so on, and it crosses even into the virology. So I think this is all I wanted to say to you this morning, and I'm ready to answer any questions you may have.
Thank you for, there we go. Thank you for your presentation, and I open it up to the floor. Are there any questions? Dr. Portnoy, first again with his question.
Great, just full of questions today. Thank you for that overview. It was very interesting. I guess one question I have, these investigators are studying products that are also potentially being regulated, and I was just wondering how you address the issue of potential conflict of interest if the researchers' work conflicts or somehow overlaps with the products that are being evaluated and regulated.
Well, yeah, it's a good question. You know, it has both ways because, of course, being an expert in this product makes you a better reviewer. But if you are invested too much in this product, that could probably raise some issues, but some people can question your impartiality. So that's why if our investigators collaborate with somebody who is involved in product development, this undergoes an internal review. So every time our scientists want to collaborate with an outside organizations, be it in academia or industrial, it goes through a review. And in some cases, the decision is made that this collaboration is possible. But then in this case, a reviewer has to excuse himself or herself from being involved. But we try to basically fine line so that this does not happen. So we try to avoid situations when it can create or be perceived as a conflict of interest.
The main thing is you have a procedure in place to address that.
Yes. We have a process. It's a sign-off. There is even a special form that a researcher has to, and it goes through the sign-off, chain all the way to the center director's office.
Dr. Assad is next, although, and then Dr. Elkins wants to be on perhaps to answer to further address Dr. Portnoy's question. But Amal, why don't you go ahead and ask your question first?
Yes, thank you, Dr. Chumakov, for a very concise presentation. My question is, what is the size of the portfolio that the OVRR holds, and is the manpower sufficient for that portfolio?
Well, if by portfolio you mean the research programs, then we have 17 research programs in this division of bacterial products, as well as 16 research programs in division of viral products. So it's overall close to 35 Some of these programs are subdivided into projects. So actually, we have one PI, one program. So each PI has a program that can be broad or more narrow. But overall, I think we have close to 90 research projects in the office.
Okay, my question was, I'm sorry, to make it clear, regulatory-wise. So, you know, continuously reviewing safety, efficacy of various vaccines. How many vaccines do you oversee? And do you have enough people to do that job?
Well, I mean, there are, I think, close to 90 licensed products, licensed vaccines, and the regulatory workload on each of them is different because, of course, there are old products where there are only amendments, basically, occasionally, and it depends, of course, on the manufacturer who submits them as necessary, but there are also products that are actively considered, such as investigational new drugs, IND applications. I don't know exactly what the number, but I suspect it's in the hundreds each year. And of course, biologic license applications is something that actually occupies, I think, most of the time of reviewers because this is a really major deal. And each year, I think there are several biologics license applications that are successfully approved. There are more, perhaps, that are still in the works. So, yeah, of course, especially with the pandemic, the workload has increased dramatically. So that really impacted some toll on our ability to conduct research. So because, of course, resources are limited, the office employs more than 200 employees, and maybe even more than 300. Each office, each division is about 100 plus scientists that are involved in it, and the full-time regulatory division is also close to 100 people. So, I mean, it's a significant number of employees, and the workload is really high. So that's the best I can answer your question.
Yeah, thank you. Thank you for clarifying.
Dr. Elkins, you had a question or comment? Is Dr. Elkins available for her question or comment?
Dr. Elkins, you're unmuted. There you go. Double mute.
Dreaded double mute, yep.
You can turn your camera on too if you like.
I can find that button. I'm obviously not doing well on buttons here. Back to the question about the studying the products that may be a conflict. I think it'd be fair to say that the vast majority of our research activities are studying classes of products, prototypes, or the pathogens themselves, for instance, in the OVRR world, not individual products that are on a path for clinical development or that are already in clinical development. That kind of circumstance is, I think, very much a minority of our research activities. And as Kostya outlined, when that circumstance arises, we have an internal process for reviewing conflict of interest, creating collaborative agreements as necessary, and recusal if necessary. But that's really the exception, not the rule. I hope that helps.
Thank you. Dr. Maliki.
Yes. So if one section of, for example, like you said during the pandemic, one group is inundated with excessive work or projects or products that are presented that have to be regulated, how is that handled in general? So are there resources to add funds or personnel to a program temporarily or remove them? Do you shift people? Do you assign postdocs? How is that handled?
I think maybe Dr. Gruber can better answer this question.
Hi, Marion Gruber. I'm Office Director of Office of Vaccines, I guess, for another 24 hours. I just wanted to answer this question. I mean, the way we handled the resources, which of course are limited, as you've heard, over the last 2 years when we dealt with the COVID pandemic and the authorization and licensure of vaccines against COVID-19 was as follows. We tried to really make use of those divisions who were not immediately involved with COVID-19 work. That was, in this case, the Division of Bacterial, Parasitic, and Allergenic Products. So we have some very experienced reviewers and also regulatory reviewers who sort of, could step in as needed to take over some of the work that the staff in other divisions could not really focus on. What we also had to do is we had to deprioritize some files and vaccines that were nevertheless critical for public health, but we just had to wait and could not really focus on our attention on these. So and then what we have done in the regulatory divisions, we have used people that would help out in the review of COVID files and sort of reshuffled and reorganized assignment. So it was really, and it still is, a juggling act. And a final most important point is people really do work long hours, canceled vacations, canceled weekends, in order to really be on top of the pandemic and the review of these files in order to cope with this workload. And that, of course, then not only pertains to people immediately involved in COVID work, vaccine work, but also other people who sort of as backup. So that's how we did that, and it is a challenging situation, and it is understood and appreciated by CBER leadership that these efforts are not sustainable. But that's the reality that we have been facing. Thank you.
And if I may also add, on the research side, before the pandemic, we did not have a single research program that would address the coronavirus issues. But immediately, right after at least 4 principal investigators immediately stepped in and started working on coronavirus, performing a really great research that helps our regulatory mission as well. So I think that historically it's been the case when our PIs responded just flipping on the dime and addressing important public health issues. So that's actually been the hallmark of our research program, a very, very high agility.
So do they have a method to, for example, let's say in that program, to if PIs, your PIs are more engaged in regulatory programs, that perhaps maybe they could be, there's a pool of funds that could give them postdocs or tags to their program going. That, I think that would probably, I mean, that's kind of what happens sometimes with the USDA, so I was just wondering if you had similar program because of the COVID and how much efforts have been put into that. I was wondering if that's one way to sustain, keep the research part functioning.
Yeah, there was some additional funding available, not really very significant, and of course during the pandemic it was not easy to expand anything, because when the labs were first month of the pandemic, our labs were practically closed, and then they started opening up gradually. So it was a really additional challenge to deal with this, but nevertheless, actually we managed to do it without significant increase in funding and staffing and so on. So.
Thank you, Dr. Maliki, Dr. Gruber, and Dr. Chumakov. Thank you for your presentation. If there are no other questions, I would ask that we move on with the presentation by Dr. Ron Raven. Ron is the chief of the Laboratory of Immunobiochemistry and will be presenting an overview of those research programs. So Ron, it's showtime. Ron, I think you're muted.
Yep, you're just muted. You're muted on your phone, sir.
Oh, how about now? How about now?
There you go.
That's
now
right.
You're there.
All right. All right. I muted my headphones and my phone. So, yeah. All right. Good. We're good. Well, first I want to thank you all again for joining us this morning for APAC. And I also want to thank you for your thoughtful and insightful questions. This morning I'm going to discuss the mission of our laboratory with regard to both the mission and the relevance of our lot release program and our research lab to the relevance of those things to the mission, the overall mission of the lab and our regulatory responsibilities. So the mission of our lab is to assure the safety and efficacy of allergenic products. We do this through the biologic license application and the investigational new drug review. We represent FDA to domestic and international academic or government organizations. We provide expert advice to FDA centers, and we monitor allergen extract potency and lot release of the standardized extracts in new products, and then we have our research program. And so first, to discuss the BLA and the INDs and our representation to outside organizations. When we represent when we perform BLA and IND application review, our lab is responsible for this chemistry, what's referred to as the chemistry, manufacturing, and control. Which means that this is all about how the product is made, the processes, and for quality and consistency. Now, I would state parenthetically that when I joined FDA, I was the clinical reviewer for these products. But when I joined FDA, we had very few actually novel products to review. Most of our INDs were mechanistic studies in which investigators were using allergenic extracts to, say, for bronchial challenge, for example, and we had to assure that the studies were performed safely. As you all, I think, as this group well understands, you know, for the good of the, you know, for the best of public health, there's been an explosion in potential products that are being studied under IND and BLA, and as that happened, I couldn't really manage both the clinical review and the CMC and supervise the CMC. And so we now have 3 really excellent clinical reviewers for this. But the CMC review is integral to the review process as an IND progresses from phase 1 to phase 3, and if so, to BLA. And then once, if a license is granted, we review the annual reports and supplements. And so it is an ongoing process throughout the lifetime of a product, as you've been told by my colleagues. We also represent the FDA to different organizations each year. Either Dr. Slater and or myself speak to the Quad AI annual meeting of the Board of Directors, and we participate on the Committee for Standardization. We have an ongoing conversation, if you will, and participate in the triennial Paul Ehrlich Institute seminars in Germany, which unfortunately, as you can imagine, one of them was postponed in the past year. I work with the European Medicines Agency quite a bit and have co-authored position papers with them on both allergic responses to the COVID vaccines and on other issues regarding standardization and testing of allergens. I also participate with the IACI, and I'm on the editorial board of 2 journals. All right, okay, there we go. So we do give expert advice to other FDA centers. When consulted, there's a formal consultation advice and process for that. And we've consulted, in particular, I've worked on consults with the Center for Drugs, the Center for Devices and Radiological Health, and the Center for Drugs again. Forgive that error. And so the other thing that we do is we have a reference reagent lab for monitoring the allergen extract potency and lot release of standardized extracts and new products. And although I need not remind the members of the committee for purposes of completion of the talk, if you will, there are standardized extracts which are controlled for potency and stability, and the potency is measured as a relative to a U.S. standard, and the unage correlates to biological potency and can vary depending upon the product as either mass units, BAU or AU, or measurement of a specific allergen. There are a myriad of non-standardized allergens in which, you know, the unitage is either PNU or weight per volume, but it's really completely, it's not informative because it doesn't correlate to any index of biological activity or immunologic reactivity. There are 19 standardized products that cover house dust mites, insect venoms, cat hair, dander, grass pollens, short ragweed pollen, and we have here that there are over 1,000 non-standardized products, and that's true. Strictly speaking, but really a lot of these products, the manufacturers do not make or market them anymore, so there are fewer than that are actually on the market. But in the process of regulating the standardized products, we have a reference reagent laboratory that is ISO 17025 accredited. And what we do is we look at lot release for standardized allergen extracts for injection. We also monitor the potency of the tablets for sublingual, for slit, immunotherapy, and we monitor the lot release specifications for the OIT product that was recently approved. We distribute, we really actually create and distribute and maintain the U.S. reference reagents for standardized allergen extracts, and this takes up a great deal of time. We have 3 FTEs right now whose sole work, actually 4, whose sole work is, this is basically almost their sole responsibilities. Again, we're ISO 17025 accredited, which requires stringent documentation and SOP protocol and annual reaccreditation. And then we're improving the methods of standardization, and we've spoken a lot about this, but I'm really pleased to say that, you know, development of an ELISA for regulatory purposes is a little different than for you know, a research purpose, and it's taken quite a bit of work, but our catalyzer is about done, and we have, you know, great CVs on a single product from plate to plate, you know, below 20%. I mean, it's really, it's going to improve, I think, standardization of this product beyond the current methods that we use. These are our staff. We have 3 principal senior investigators, myself, Dr. Slater, and Dr. Alexander Zobner, who was brought on within about a year and a half ago. We have staff fellows and scientists. These are FTEs, Dr. Hillier, Dr. Levinson, Dr. Strader, and now Dr. Zhuifei Ma, who joined us just last July. All of these 7 people participate in the review process, as well as the reference reagent people, colleagues in that lab listed on the right. So lastly, I'm going to just discuss our research project very briefly. And I think you all know that for immunotherapy, these are complex natural products. They're unselective aqueous extractions of natural source material, diagnostic products, and they're diagnostic and therapeutic products as well. Allergic disease is complex. I think I don't need to tell you that. It involves genetic predisposition, multiple organ systems, innate adaptive immunity, viral infection, microbiome, and so on. The future of allergen therapy is for targeted populations, viral-like particles, shorter intermediate length peptides, complex mixtures for oral immunotherapy for food allergies, immunotherapy, adjuvants, DNA vaccines. All these things have been reported in the literature. I'm not telling you anything that is disclosing confidential information by telling you about this. So in understanding these things, then we need to consider mechanisms, including the protein chemistry of a lot of these products and the innate and adaptive immunology. And to that regard, those are the things that we study. And so the Slater lab in particular studies allergen protein structure and focuses primarily on indoor environmental allergen. We study research on viral respiratory infections and interferons and how allergens affect the antiviral response. So we share a common interest in these environmental allergens. And Dr. Zhovner's lab is focusing on food allergy. And so, we all are interested in the innate and, for him, the adaptive mucosal response to allergens. So with that, I will take questions. And thank you very much for your time and your attention.
All right. Thank you, Ron. There are questions. Dr. Nolte is first with his question. If you'll...
Thank you very much, Dr. Rabin. Can you hear me okay?
I can, and I can see you.
All right. So I have 2 questions. One pertains to your mission statement to assure safety and efficacy of allergenic products. And you mentioned that there are maybe about 600 extracts which are commercially available in the U.S. which are unstandardized. Are there any plans to try to move on with further standardization, like tree pollen is a very common allergen?
Yeah.
Because I wonder how we can assure safe and efficacy use of uncharacterized, unstandardized extracts according to modern standards. That's my first question.
Thank you for that question. So yeah, there are. The issue is, Dr. Nolte.
Hello, this is Connie with Senior Aid Helper speaking on a recorded.
Okay. The issue with that is that the mechanism of standardization has generally been, serologically based or antibody based in the past, and we kind of recognize that has huge limitations with regard to reference replacement, with regard to precision, and these are really late 20th century techniques. Dr. Slater's lab is really focused on defining products using mass spec and using, a tandem mass spec. And I think this is, we and the European groups, particularly Paul Ehrlich, really believe that this is integral to the future of allergen standardization, because although the development can actually, once we get it down, can actually take less time per product than developing immunological reagents and so forth. Once that development is down, then reference reagent replacement is trivial. and that can be expanded, you know, greatly. And so really, that's really what we want to do. And we've done some preliminary work. We've got some great work that was done with house dust mite to sort of overlap with what we know about house dust mite. Dr. Slater is working on alternaria right now as one indoor allergen. And this is really where I am hoping to take this, because, you know, for tree pollens, for example, you know, once you've identified, you know, there are, I mean, all these, most of these major allergens, they're characterized, right? We know the oak pollen allergens, we know maple and elm and birch. And once you've characterized them and you can pull out signature peptides and do orthogonal tests for, you know, for degradation and so on, you can apply these to whole families, right? I mean, you know, white oak, red oak, and so on. And that would be the idea. So instead of developing, you know, one assay and then looking for cross-reactivity, you know the genome of these. And so we're very excited about this project. And,
thank you very much.
So that's
you're welcome. Other question I have is, in the industry, often we study very carefully the FDA guidance on drug development, and there's clearly a lot of interest around developing drugs or treatments for food allergy. Is the agency planning to come up with an immunotherapy development guidance from the FDA, as they have done in other countries?
So the short answer, no. The answer is no. There are no immediate plans for such guidance document. But I would say that, you know, based on our experience with Palforzia, we certainly have a template. But like everything, you know, the first version is the beta version, and we understand that, you know, now we know what we're looking for. We know how to do it. But as far as putting that out in a guidance document, there are no current plans.
Okay, thank you.
Anyone else?
Yes, you have quite a coterie here. That first one is Dr. Greenberg. Dr. Greenberg actually was the first on the list.
Yes, sir. All right. There we go. How are you? You're muted, sir. You're
muted.
Sir.
Sorry.
There we go.
Thank you for the presentation, Ron. Might you point out the collaborations with the Paul Ehrlich team, because I think that's quite important that my understanding, you're working together. I know you are, but I think that,
a little bit.
At least I'd like to get an update on that. And a second question was on staff turnover. I didn't hear that presented by anyone yet this morning. And the third is external allergen expertise, like for alternaria you mentioned, but or cladosporium. Are there programs working on the fungi that could be informative or helpful? The first was about Paul Ehrlich.
The first was Paul Ehrlich. So I would say that collaboration has waxed and waned a little bit, and it has certainly waxed with the pandemic. And it's kind of odd because they were the first ones to pick up with the mass spec, and you know they were ahead of us. But you know right now, especially with bringing, we had two really very skilled chemists working on this. First, Dr. Sam Mindaye, who did leave the group about 2 or 3 years ago, but now Dr. Brad Strader, who's just a fantastic, you know, skilled at MS. and protein chemistry, and I think we're actually kind of leapfrogging them a little bit. But at any rate, I think that we're picking up the collaboration. There's nothing like as you well know, these science by Zoom has limited opportunities, and my guess is that the next time we see them, that the collaborations will pick up some strength, because we did have someone who actually kicked off the house dust mite program who was here for a couple of years, Dr. Yelena Spirik, and then now she's at Paul Ehrlich. So we do have a real opportunity to strengthen that relationship, and I think that, I know that Dr. Slater and Dr. Strader have been in contact with them, and we're really looking forward to doing that. So that was your first question. Your second question, I'm sorry, remind me.
Turnover. What are you doing?
So
what
turnover, so I
issues?
referred to that a little bit. So in the reference reagent lab, we have very little turnover. I mean, it's been remarkable. We had a Dr. Lindsay Wren who began the quality assurance program as the quality manager. And if you're familiar with ISO 17025, it's kind of a specialized position, because the QM does not participate in the lab work themselves. They oversee the lab work and cannot, and have to do that from a distance. That is according to the ISO 17025 regulations. Dr. Wren also performed some research, and that was a bit overwhelming, and she decided that she wanted to go into the regulatory realm, and so she left us for CBER, and we ended up sort of expanding our group in the process, and now we have Dr. Zagorski, who is really an excellent quality manager, and spares me a lot of work. I mean, she's a godsend. And then the other three people have been in the reference reagent lab, have been in there, two of them have been in there since I joined the agency, and the third one has been, Aaron has been with us for 15 years. On the research and regulatory side, we've had some turnover with Dr. Wren leaving. Dr. Hillier has been with me for 12 or 13 years. And we've added new people with a little bit of turnover, but I think we don't have a great deal. But right now, the biggest issue is having everybody on board who can do the regulatory work. Now, Dr. Strader came from a different office, so he's a very skilled regulator. But allergenics are different from blood products, which is what he did before. So, but he's bringing up to speed. Dr. Levinson is learning the skills, the reviewer skills. Dr. Hillier is an extraordinarily skilled reviewer, and so she's teaching doctors, the others who are coming on. But they seem to be happy here, and so hopefully we'll have less turnover. I'm going to leave your last question to Dr. Slater.
Okay, thank you.
Dr. Davis, you're next.
Wait. Should Jay answer his question?
If you have a direct answer to that, yeah, and then we'll go back to
Just ask the question again, Dr. Greenberger. I'm sorry.
The question was on expertise, like laboratory expertise regarding fungi.
Yeah.
Like I know you mentioned Alternaria, but like Cladosporium, is that a priority?
We're absolutely interested in that. I think when Dr. Strader and I got farther into looking at Alternaria and the things that we were finding initially, it rapidly became apparent to both of us that we needed to expand our biological expertise on fungi. We've now established a collaboration after some searching with a really, really good group in Portugal with whom we're collaborating. They have a deep interest in the biology and immunology of Alternaria infections as well as allergy. And I think our expertises really sync very, very nicely. So I'm very optimistic about that. Ron, if I could take just 1 minute to scoot back to a quick answer to to Dr. Nolte's question, if that's okay with you, Dr. Peden, very, very quickly. You know, as our abilities and our technologies and our insights into biology and pharmacology improves, FDA's always looking back over its shoulder at products that we approved a year ago, 10 years ago, and decades ago, and wondering whether these products are really up to modern standards. When there are clear safety and efficacy issues, we are capable of revisiting this, but as a general practice, we don't find that to be the case. And I have to say, we are very much committed to improving our standards for allergen extracts and allergenic products in general, and we will continue to do that going forward. It does not mean that the existing licensed products aren't safe and effective and branded properly as they are currently licensed. It's very easy to go back and say, well, these products are by 20th century standards. They are licensed products, and they are good products, and we have, in just the past few years, taken products off the market where we've been concerned about their safety. So I just want to make sure we don't slip down the slope of saying, well, everything that's not up to the standards of post-1997 are inferior products. They are a class of non-standardized products. We recognize that, but they are safe and effective when used as indicated. And then I'll be quiet. Thank you.
Thanks for that update and clarification. Now, Dr. Davis, thank you for your indulgence. It's your turn.
Thank you very much. So really nice presentation, Dr. Raven. I just have, yeah, good to see you as well. I actually have three questions. One is, I'm quite impressed with the work for the indoor allergens in terms of the mouse and fungal cockroach work. But there is one allergen that's pretty significant from a subclinical standpoint, and that is, oh, and also you included cat. So is there any thought into addressing dog allergies? As you use very frequently, there are some concerns potentially about efficacy. Is there any plan to delve into that?
I'm going to refer that to Dr. Slater because I think he's got a great answer, and it's his work.
Yeah, Dr. Davis, thank you for that question. We did actually start looking at dog several years ago. We pursued it to a certain degree, and then we swerved away from it when we found other allergens that seemed to be giving us more interesting data. I couldn't agree with you more. I think, in fact, is my dog here? No, she's not here. But I couldn't agree with you more. I think this is a very, very important area for investigation. And frankly, you know, Ron was talking up how exciting it is to have great people on the mass spec beat in our lab. I think we are going to be turning to dog to look at it much more carefully. In fact, I think it might be a very good complement to our current work, which has really focused on enormously complicated allergen systems, mouse, alternaria. These are wildly complicated, and it might actually be a great thing to do to look at dog, which I think in the end is going to be relatively simpler, but not quite as simple as we think it is. So I agree with you. Thank you for bringing that up.
Yeah, no problem. So my second question, I have a couple more questions. So my second question is actually in terms of the mass spec evaluation of environmental allergens. It definitely, I'm excited to see that that there is movement towards making that, you know, something that can be utilized more widely. Is there plans? Because I really sympathize with Dr. Nolte's thought about the usefulness of guidance for food allergenic product development. Is it what is the agency's thought process regarding the aspect surrounding food allergies? And is that something that you see as becoming more widely available?
I do. I do. Whether or not it would be in the context of a guidance document, I definitely do. Okay. I think it's really important, and I think it's an extraordinarily valuable tool. And I can't speak to specifics, but I can tell you that it's always on my mind when we see these products, and we really do understand the value of this tool.
Mm-hmm. Yeah, it might be useful, I think, for everyone in the field really to understand the importance of it.
Yeah, so the only thing that I can say is we've written a number of reviews, and we're writing one now, and whenever we write them we include that, and it's sort of included at the end of the review, and you can look at our published papers. My name is on all of them, and Dr. Slater's, we're both co-authors on most of them, and so on. And we, and it's not just sort of a lip service thing that, we're just saying, oh yeah, this would be cool. I mean, we really are thinking about it in a very, very important way.
Okay, great.
Your third question, please.
My third question relates to this idea of this balance of regulatory activities and research. I did notice that with the exception of probably Dr. Hillier, who's really skilled, the routings of INDs, the ones that have the highest routings, are not with the LIB anymore. So I'm just wondering about, how the routings are, the number of routings are decided, and if there's some consideration of equity just to make sure that, and I don't know if that has anything to do with.
So are you referring to which INDs that we get for review, which ones are placed when you say routings? So that's actually done sort of beyond our pay grade. They come to us, and most of them are pretty obvious, but there are some that we kind of dispute because we want them, and there are some that we can dispute because we don't want them. I mean, you can imagine that sort of thing goes on. And but in general, we get what we get, and we don't get what we don't get. And then, as far as how it works out in time, I think Dr. Elkins really described it pretty well. For us, there's a lot of feast and famine, so we'll have a lot of time in which we can really focus as senior staff on the projects, and there are times that we don't, on which the review work really, crushes us a little bit. And I don't think we've been, I'm certain that we have not been, things haven't been rough for us as much, as for the COVID people. So we're helping them a little bit, trying to relieve their burden. And certainly when we had those 3 split products, things were pretty crushing for me. But more to the point, we do have, I didn't tell you this, but we do have post-bac research assistants who come and work with us. And as you well know from your own experience, you get some real superstars in there, and who you can direct and say, this is what you're doing for the week, and that what they're doing for a week. And so the projects don't, hit a complete dead end by any means when we're nailed with the review work.
I hate to interrupt. We do have
running out of
3
time.
more questions, and we have about 70 seconds.
I'll try to be very brief. I'll try to be very brief. Sorry.
So, Jay, you have a question?
A little loquacious today.
Jay.
Oh, I think.
Yes. Right. It caught me by surprise. I thought there were more people in line. Quick question.
There are, but
right
they're behind you, Jay.
Huh? Okay. Quick question about components. I can do a laboratory test to measure components now for the major allergens in a lot of these extracts.
Yeah.
Has there been any thought to asking the companies to label the concentration of those components in their extracts so that I can more rationally select which extracts to use with my patients?
The current rules are that we define the methods of standardization, and I think that, you know, we are having, you know, I think, and right now, unless you have a major allergen, the methods are not to label single components. But certainly the idea of the mass spec is to do something like that, and I'd rather not comment on that. That's all I'd like to say for the interest of time.
And is your lab involved in licensing diagnostic tests, or who does that? Because the
No,
tests are
that would
licensed
be
by the
CBER.
FDA. How's that?
No, that would be CBER. That would be CBER. I mean, they, you know, as I said, yeah. So let's go on to the next, please. I'm sorry.
Dr. Maliki?
Hi.
Hi, how are you? Good morning. I've got a couple of, maybe a comment and a question really. One, that we know that there's like a massive amount of variation, significant, clinically and relevant and from a biological or chemical standpoint in skin test extracts. So, and I don't know how heavily they're scrutinized, but there is a very significant difference where you purchase it, how the extracts look, how they immunologically react. So I'm assuming that with more immunotherapies and so forth, BLAs coming out, or that these, and this could be a yes and no just to save time and we can talk later, but there's going to be much higher scrutiny of these extracts when it's coming to use as immunotherapy. The one comment I have is that you mentioned something about like the immunological techniques or like old school or, you know, doing ELISAs. Well, obviously, you know, being a biochemist, molecular biologist that studies the structure and function of allergens, that we know that one, that if you go to a peptide-based method, you may miss one structural epitopes.
Yeah.
So the peptide could be there, but your extract could be not reacting.
Yeah.
Binding and the immunological responses change.
Yeah.
So that kind of, so I think it's got to be some type of combination of things.
Oh, absolutely. And I'm sorry I didn't make that clear, but there will always be an orthogonal test. You know, after all, this is immunology. So let me reassure you of that. I think we have one more question, so let's just do that real quickly.
We actually have two more people with their hands raised, and we have a little bit more time if there are other questions. If not,
all right.
Dr. Woodfork and Dykowitz, and then we'll go to break after that unless someone has another question.
Dr. Woodfork, how are you? Nice to see you.
I'm very good. Mine is just a general question around the flexibility that the program has in terms of your future research directions. We've already talked about all the multitude of areas. There's some in the report around glycosylation. Can you comment very briefly on the flexibility you have with regards to the priorities and the future scope of research and potential for expansion?
So the potential for expansion is relevant to space considerations and money considerations. We're kind of filled up with space and, you know, for money. We've had good years and bad years, you know, but there are there are two mechanisms, as you can imagine. There are FTEs that people fight over, and then there's using soft money, you know, type mechanisms to bring postdocs and postdocs in. So, you know, everybody has different flexibility depending upon how much money you can get. With regard to, you know, being nimble about the research, well, you know, I study viral illnesses, right? And, you know, I study RSV and RV, and, you know, one could say, what are you doing that for? You're not in DVP, but it's related to asthma, and I've been given freedom for doing that. So I think if you can really relate it to the mission of your lab, we've got pretty good freedom. But, you know, I think we have to do that, obviously. This is taxpayer money, and the whole argument is that by studying these things, we're, enhancing the regulatory mission of the lab. But it's been, I can't complain. Let's put it that way.
Thank you.
You're welcome. Last question?
Dr. Dykowitz.
Yes, sir. You're muted, sir.
You're muted.
There you go, sir. I got it.
All right, thank you. My question is, in a larger sense, Dr. Slater's lab is doing great work looking at the heterogeneity of allergenic extracts, both between products and lots. But then there's also the issue in terms of the diagnostic utility of the extracts that are being.
Right.
And so how do we get from an assessment of the heterogeneity of composition to application?
I think it's moving towards some sort of a component-based technique. And, I think that the way that we're approaching it is with a mass spec, but I think that I think that's the bottom line. The bottom line is component-based technique, and that's where we really want to go, because we know that there's some allergens that are dominant, and there's some based on genetics and so forth. But that's the bottom line in a nutshell. We are thinking about that.
Good. And I guess I think from standing forward, though, for, again, clinical utility, do we envision that, for instance, manufacturers of particular extracts might be internal data about the diagnostic utility? Is that something that the agency...
I'm going to put my, in closing here, I'm going to put myself, on a little bit on a limb here and say that, my little dream for things is, as I, wrap up my time here, is that if we're able to define techniques by which manufacturers could, do a component-based characterization of their extracts, then the market will, will push them towards, developing better extracts. And that's, I sort of think that's kind of, the American way of approaching things. And so, that's my dream. So I hope that answers your question.
Yeah, thank you.
All right. Well, thank you all for your patience and for your very interesting questions and for your engagement. And I think we're all ready for more coffee or lunch. Yes, Dr. Peden.
Well, if you need a 10 minutes lunch, that's great. 10 minutes. So we have a 10-minute break. It's 11:18. Let's just call it 11:30. So you're not. We'll be back at 11:30.
All right.
My dogs will be back then too.
All right. Hold on one second. So at this time, we will take break. Studio, please put us to break. And captioners during break, please. All right, welcome back from that break. And again, this is the 31st meeting of our APAC meeting. I'll hand it back to Dr. David Peden to continue us on. Dr. Peden, take it away.
We're now supposed to go into the open session. However, as there were no formal requests for oral presentations from the general public, we will now be moving to closed session.
Thank you, Dr. Peden. So everyone just wait a moment while we move into the breakout room for the closed session portion of the meeting, and the public portion of today's meeting has concluded. So just a note for captioners and anybody viewing the meeting virtually, the portion has concluded.
All right, so thank you much, everyone, and I will now initiate our closed portion of the meeting. Here we go.
Sub-conference rooms are open.